The next thing we're getting into with the membrane, after covering phospholipids, cholesterol, we've looked at what a liposome and a micelle is and the bilayer, and the concept of fluidity, or how flexible that a membrane can be and how it's affected by temperature, but it's also affected chemically through the tails and through the cholesterol molecules. If you didn't get that, go back and look at the other material on those topics. Now we're going to keep moving forward. So a membrane, which is starting off here with the phospholipids and cholesterol, still has more parts to it, more components to it. There are proteins. The proteins we find can make up a really large part of a membrane. Some membranes could be 80% of the membrane is protein. All right, so it could be a huge portion of the membrane. It's not just phospholipids. But this is the core of it. That's what kind of forms the membrane. It starts off here, even though proteins can be in great number. So what are these proteins? What do they do? How were they kind of discovered? So initially what people found were proteins called peripheral proteins. So I'll write these down here. Peripheral proteins. So peripheral proteins are attached to one side of the membrane. So we have alpha helix, beta sheet, and the peripheral protein is embedded typically in the membrane like this. Now the peripheral proteins can be attached sometimes to the lipids. They could be stuck here within the membrane, but they're typically only on one side. They don't go all the way through. That's, I got a space, peripheral. We'll get into the jobs of many of the peripheral proteins, some even specific names of some peripheral proteins. But they only occur on one side, but they could occur on either side. So we can have peripheral proteins on this side, or this side. So there could be peripheral proteins on either side of the membrane. That's peripheral. This one's peripheral. It's not only one side of the membrane. They could be on either, but they do not travel all the way through. And then there are membranes that, then there are proteins that we call transmembrane, or also referred to as integral protein. So it kind of depends on where you look, which particular book or website or other piece of information you get. Transmembrane protein is my preferred term simply because it's more descriptive. We'll find in this course, as in a lot of biology, there are often multiple terms for the exact same thing, which can be very confusing at times, especially for new students who are just starting to learn. My preference is to use the term then that is the most descriptive, and also use the term that is the most well used in the scientific field. We'll find sometimes in textbooks the authors uses a term, but scientists don't use that term. They never say that term. So I'll try to present both sides of it. And here I'm saying transmembrane protein is a protein that is going to span the entire membrane. It is transmembrane. So we have this protein on one side of the membrane, but then it goes all the way through the phospholipid bilayer, and we see some of the protein on the other side of the membrane. So this one here is what we call transmembrane. Now, because it's embedded in the membrane all the way through, it's where it's also called integral. So it's integral, it's integrated, into the membrane itself, and part of it. A couple things to note. A transmembrane protein has this section that spans the membrane that actually goes through the fatty acid tail region. We're not going to get into all the detail, of the chemistry of it, but a few things that we could point out. One, that section is almost always, I don't know if you recognize it from my squiggly drawing, from secondary structure of proteins, it's the alpha helix. So that's an alpha helix structure that goes through there. Almost always, it'd be an alpha helix structure. In addition to that, the R groups of the amino acids, remember, if you go back to amino acid structure, each amino acid has a central carbon, has an amino group and a carboxyl group, all of them. And then there's an R group. It's what makes each amino acid unique. Some of them are nonpolar. It's carbon and hydrogens. Those are ones you typically find here. You find them in the region of the tails, which are also nonpolar. So they associate with each other. They kind of stabilize each other, that sort of thing. So these are just terms right now and just giving you an overview. We're going to get into, again, functions. There's all kind of functions of transmembrane proteins that we'll get into shortly. But for right now, what we're going to find is just structurally we have phospholipids, cholesterol, and now we have two types of proteins, some that go all the way through and some that are just attached to one side, peripheral or transmembrane. All right, and then again, we'll get to function in a little bit. And so we got these guys checked off. Now we're going to look at glycoprotein, glycolipid very briefly before we get into function of the overall membrane, as a whole. So you might get from these names here, glyco is part of both of them. Glyco means, if you remember, sugar. Okay, so these are sugar molecules, typically in long chains attached to the surface of the membrane. Now those sugars, we can attach them in these chains. I'll draw a little branching chain, like this. So this is a sugar chain. Sugar chain, that's just a real simplistic terminology here for what we have, but it's a polysaccharide. It's a bunch of sugars attached to each other with glycosidic links. And then in this case, I drew it attached to this polar head. So as a result, this sugar, this glyco, is attached to a lipid. So we call that a glycolipid, a sugar lipid. We can also have a sugar chain, let's say, attached here to this protein. This sugar chain attached to a protein is now a glycoprotein. So the glyco is the sugar chain that's attached. And then it can either be attached to a protein, like a protein, could be attached to one of the phospholipids, is a glycolipid. Now, the thing to note about these, the glycoproteins and the glycolipids, they are only on one side. They are only on the outside. They are extracellular only. All right. Unlike the peripheral proteins. So I'm going to erase this term from here so we don't get confused here. Peripheral proteins, which I'm erasing there because I don't want it near that term. Peripheral proteins could be on either side. Peripheral proteins could be inside. So let's say this is the cytoplasm of the cell. And let's say this environment up here is extracellular. Okay, outside the cell. So this is the outside of the cell. This is the inside of the cell. Peripheral proteins can be on either side. Glycoproteins are extracellular. They're only on the outside of the cell. We do not see glycoproteins attached to the inside. So what this means is that people can take a live cell, break it, get a chunk of membrane. It would be a phospholipid bilayer. That phospholipid bilayer would have proteins associated with it. Proteins on both sides of it, proteins going all the way through it, cholesterol molecules embedded in it. But if they were to look at that little piece, chunk of membrane, and say, which side would have been outside the cell, which side would have been inside the cell, it's easy to tell. The outside surface is covered in the sugars. The inside is bare, no sugars inside it. It can have proteins attached to it. There's sugars in the cytoplasm, but they're not attached to the surface of the cell. That's important, because it's related to their function. Glycoproteins and glycolipids, their job is often in cellular identity. So one example of this are things called MHCs. So this is major, M, histocompatibility, H, C complex, MHC. So this is why we say MHC and not major histocompatibility complex all the time. MHCs are glycoproteins, glycolipids. These are, for example, the molecules that give you your blood type. So if you're type A blood, you have these type A glycoproteins attached to the surface of your red blood cells. It is the sugars that give them the particular identity. Which are these sugars, the order in which the sugars are, the branching nature of the sugar. So these are the unique characteristics of that particular glycoprotein. And then cells in your body will recognize those sugars coding your red blood cells as being belonging to you. If you were to get type B blood, it would be a different group of sugars. Your body would recognize that as being foreign. It would say, this is not me, this is something else. So it's also a way to recognize self. So your cells all have your own MHC markers on them that are unique to you, not just for blood type, but for every single cell in your entire body. And it's very unique. This is what makes tissue and organ transplant difficult, because all cells are labeled unique to individuals, and this is how they're labeled with these sugars. It's not going to be a big part of what we get into in this particular class. We're going to talk more about membrane function in terms of transport, things moving across the membrane. We talked very briefly a little bit at a few times about the sugars and MHCs. Right now, we're just breaking it down into structure. So we're listing the molecules that make a cell membrane. They include the phospholipids, the cholesterol. So we have a bilayer with cholesterols. We then have sugars. They're only on the outside of the cell. Okay, they could be attached to a protein, glycoprotein. It could be attached to a lipid, glycolipid. And then the proteins themselves, some of them are spanning the entire membrane. They go all the way through their transmembrane, and some of them are attached to only one side. They're peripheral. The ones that are attached to only one side, though, could be on the inside or the outside. Okay, not the same protein, they're different proteins, but you can have peripheral, the general category of protein, on either side of the membrane. And that, at this point, is the breakdown of the basic structure and a few little characteristics of the membrane. So I talked a little bit about fluidity, and we talked a little bit about cell identity, just with the MHCs and the sugars, but that's pretty much it. The main function that we're going to get into now has to do with transport or movement across the membrane. So I'll introduce it, and then we'll get into it in the next lecture. Cells here are the cell membrane itself is called a selectively permeable barrier. Okay, there's no abbreviation for that. It's not like SPB, it's just selectively permeable barrier. That just means a couple things. One, it's a barrier. Okay, so that means if we have, let's just say, an environment here, and this is a membrane down the middle. So this is just a made-up scenario. Say in a lab, we have a chamber, a container, and we fill it up with water. So we put water molecules on both sides of it, and then a membrane down the middle, separating it into two halves. And then we were to put into those halves different things. Maybe in this half we add certain molecules. And let's say they're sugar molecules. The question would be, will those sugar molecules move across to the other side, or will they stay stuck on this side of that membrane? So the membrane, we're saying, is a barrier. That means it's going to prevent certain things from getting across. But we also say that the membrane is permeable. Permeable is, say, like a screen, a window screen. A window screen is, in a way, a selectively permeable barrier. It allows gas molecules, oxygen and carbon dioxide and nitrogen, to move back and forth through it. Other small particles that may be smaller than the screen's grid. So if a window screen is made up of a grid like this, and you have little particles that are smaller than the openings in the grid, they can get through. But if you have larger particles, dirt, maybe some insects, they try to get into the screen. It's a bee, he's kind of looking to get through, and he can't get through because he is larger than the screen. So the screen is selective for size, right? So only certain things can get through. A cell membrane is going to be a bit like this. The phospholipids, remember we said, There's hydrogen bonds between the heads. There's no bonds between the tails. So there are spaces, essentially, between the membranes. So something could go from one side of the membrane, right in between the phospholipids, and move to the other side. So the barrier that is preventing movement of certain things could be permeable to others. So gases, for example, can get across membranes like this little drawing. So oxygen, for example, could do this. Oxygen could just move right across a membrane from one side to the other. Oxygen molecules going between the tails of the phospholipids, between the heads, from one environment to another, in either direction, depending on concentration, which we'll get to in a second. So part of the material we're going to get into next is about the structure of the membrane itself, okay, and about the size and shape of the membrane itself. But the other aspect of what we're about to get into has to do with energy, which is something different from everything we've talked about so far. And the concept of energy and storing energy, stability, things moving from one environment to another, being driven by forces which either have them being less stable or more stable. And so that's something new that we have to get into really before we address the next part. All right, so I'm going to end this here with that. That's the structure. You should be able to sketch something like this and label. So you should sketch. So practice this. Take a blank, so copy it a few times and label it, and then take a blank piece of paper and then sketch a membrane. So start off with phospholipids. Add in little cholesterols, put little zigzags, for transmembrane proteins, put little squigglies for, peripheral proteins. Make sure you add in the sugars, but they're only on one side. Label everything. And if you can do that, that's the first step. You need to be able to do that. The next step is we're going to get into is the permeability aspect of it, and that's going to require a few other things. So we're going to cover that next.